Assalamu alaikum. Today we want to talk about chapter 18, storage mechanisms and control in carbohydrate metabolism. It will be in, in two parts. In part one, we'll talk about how glycogen is degraded and produced, and then gluconeogenesis produces glucose from pyruvate. Now, when we digest a meal high in carbohydrates, we have a supply of glucose that exceeds our needs. So we store glucose as a polymer called glycogen. Now, glycogen is similar to the starches found in plants. That's why it's sometimes called animal starch. Now, in the degradation of a glycogen, several glucose molecules residue can be released at the same time, one from each end of a branch rather than one at a time, as it would be the case in a linear polymer. Now, studies show that glycogen is optimized for its ability to store and deliver energy quickly and for the longest amount of time possible. Now, and the secret for that is optimization is the average chain length of the branches, 13 residues. Now, if the average chain length were much greater or much shorter, glycogen would not be an efficient vehicle for energy storage and release on demand. Now, glycogen is found mainly in the liver and muscle. The release of glycogen stored in the liver is triggered by low levels of glucose in the blood. So liver glycogen breaks down to glucose 6-phosphate, as we see here, and then hydrolyzed to give glucose. Now, the release of glucose from the liver by this breakdown of glycogen replenishes the supply of glucose in the blood. However, in muscle, glucose 6-phosphate obtained from glycogen breakdown enters the glycolytic pathway rather than being hydrolyzed to glucose and then exported to blood stream. Now, three reactions play roles in the conversion of a glycogen to glucose 6-phosphate. Now, in the first reaction, notice that here each glucose residue cleaved from glycogen reacts with phosphate to give glucose 1-phosphate, as you can see here. And this cleavage reaction is known as phosphorylasis rather than hydrolysis. So catalyzed by enzyme called glycogen phosphorylase. As you can see here, no ATP is hydrolyzed here. Now then, glucose 1-phosphate is isomerized to give glucose 6-phosphate, as you can see here, catalyzed by the enzyme called phosphoglucomutase, where the inorganic phosphate at anomeric carbon 1 is transferred to carbon number 6. And this is called isomerization, but by mutase. Now, complete breakdown, however, now since the glycogen phosphorylase cleaves the alpha-1,4 linkages in glycogen, complete breakdown requires an enzyme called debranching enzymes that degrade the alpha-1,6 linkages. Now, when glycogen rather than glucose is the starting material for glycolysis, there is a net gain of 3 ATP molecules for each glucose monomer rather than 2 ATP molecules as when glucose itself is the starting material. So we conclude that glycogen is a more effective energy source than glucose. Now, the debranching enzyme involves the transfer of a limited branch of 3 glucose residue, as you can see here, to the end of another branch where they are removed later by glycogen phosphorylase. Now the same debranching enzyme then hydrolyzes the alpha-1,6 glycosidic bond of the last glucose residue remaining at the branch point. Now, when an organism needs energy quickly, glycogen breakdown is important. Muscle tissue can mobilize glycogen more easily than fat and can do so anaerobically. Now, with low intensity exercise such as jogging or long distance running, fat is the preferred fuel. But as the intensity increases, muscle and liver glycogen becomes more important. That's why some athletes, middle-aged distance runners, etc., try to build up their glycogen reserves before a race. How? By eating large amounts of carbohydrates. So remember here that the debranching enzyme in its way cleaves an alpha-1,4 linkage and forms a new alpha-1,4 linkage here, in addition to hydrolyzing the alpha-1,6 that is here. Now, glycogen production. How is a glycogen formed from glucose? Now, the formation of a glycogen from glucose is not the exact reversal of the breakdown of glycogen to glucose. Why? Because the synthesis of a glycogen requires energy which is provided by this UTP. Now, in the first stage of glycogen synthesis, glucose 1-phosphate reacts with UTP to produce uridine diphosphate glucose, or the so-called UDP glucose, and also produces pyrophosphate, PPI. This is catalyzed by the enzyme called UDP glucose pyrophosphorylase. This is the structure of the UDPG molecule, which acts as a carrier of this glucose units so that it is transferred to an already existing chain. Now, if we look here, the formation of UDP glucose has delta G naught prime of 0, whereas the hydrolysis of pyrophosphate produces 7.3 kilocalorie. So what happens is that these two reactions are coupled so that the net delta G naught prime is also minus 7.3 kilocalorie. So the coupling of these two exergonic reactions to a reaction that is not energetically favorable allows an otherwise endergonic reaction to take place. And this is the idea of metabolism, is coupling, where the energy released from exergonic reaction drives endergonic one. Now, the addition of UDPG to a growing chain of glycogen is the next step in glycogen synthesis. Then UDPG is added to a growing chain of glycogen. Now, each step involves formation of a new alpha-1,4 glycosidic bond catalyzed by the enzyme called glycogen synthase. Now, this enzyme cannot simply form a bond between two isolated glucose molecules. No, it must add to an existing chain with alpha-1,4 glycosidic linkages. Now, the initiation of a glycogen synthesis requires a primer. Now, at the heart of every glycogen molecule, there is a protein or enzyme called glycogenin. It is the primer. Since this glycogenin molecule acts as a catalyst for addition of a glucose until there are about 8 of them linked together. At that point, glycogen synthase takes over. Then, glycogen synthase always adds to an existing chain of at least 8 glucose residue length. Now, synthesis of glycogen requires the formation of alpha-1,6 as well as alpha-1,4 glycosidic linkage. So we need the so-called branching enzyme accomplishes this task. It does so by transferring a segment of 7 residues long from the end of a growing chain to a branch where it catalyzes the formation of the required alpha-1,4 glycosidic linkage. As we see here, no, this figure shows us the how glycogen phosphorylase adds one glucose at an existing chain here. So UDP glucose, UDP acts as a carrier of glucose that is added to a growing chain. Now, notice here then the branching enzyme, what it does. Note that this enzyme has already catalyzed the breaking of alpha-1,4 here in the process of transferring the oligosaccharide segment. So breaking alpha-1,4, transferring this 7 glucose residue long from the end of one branch to another to form another branch. Now each transfer segment must come from a chain, this chain, at least 11 residue long. And in order to remove 7, you need this chain length to be 11, at least 11 يعني, residue long. So, and then it forms the alpha-1,6 linkage here, the new one, and each new branch point must be at least four residues away from the nearest existing branch point. So the distance between the old branch and the new branch must be at least 1, 2, 3, 4 residue from each other, as you can see here. Now, balancing glycogen breakdown and production. How does an organism ensure that glycogen synthesis and glycogen breakdown do not operate simultaneously? Because if this were to occur, the main result would be the hydrolysis of UTP, and this would be waste of chemical energy stored in the phosphoric anhydride bond. A major controlling factor lies in the behavior of glycogen phosphorylase. Now, this enzyme is not only subject to allosteric control, but also it is subject to covalent modification. You remember we saw earlier an example of this kind of control in the sodium-potassium pump in section 8.5. In that example, phosphorylation and dephosphorylation of an enzyme determine whether it was active, and similar effect takes place where. Now, this figure summarizes some of the control features that affect glycogen phosphorylase activity. أول شيء, the enzyme is a dimer, as you can see, that exists in two forms. The inactive T or TOT form, as we saw in chapter 7, and the active R for relaxed form. Now, in the T form, it can be modified by phosphorylation of a specific serine residue, each of the two subunits. Then, phosphorylation, we need 2 ATP to phosphorylate the dimer at 2 serine residue here to form the phosphorylated enzyme. Now, this esterification reaction requires an enzyme called phosphorylase kinase, and the opposite process is called dephosphorylation, and it requires the enzyme called phosphoprotein phosphatase. Now, the phosphorylated form of glycogen, phosphorylase, is called phosphorylase A. So, it is the active one, the phosphorylated one, and the dephosphorylated one is called phosphorylase B. Now, the switch from B to A is the major form of control over the activity of phosphorylase. Now, what happens in the liver, for example, here in the liver, glucose is an allosteric inhibitor of phosphorylase A. It binds to the substrate site and favors the transition to the T state. This shifts the equilibrium to the phosphorylase B. Now, in muscle, however, the main allosteric effectors are ATP, AMP, and glucose 6-phosphate. Now, when the muscle uses ATP, AMP builds up. The increase in AMP stimulates formation of the R state of phosphorylase B, which is active. Now, when ATP is plentiful or glucose 6-phosphate builds up, now these molecules act as allosteric inhibitors, shifting the equilibrium back to the T form. So, to summarize, we have two controls. We have the covalent modification, which is the phosphorylation, and then we have the non-covalent or allosteric control, the effect of glucose, AMP, ATP, and or glucose 6-phosphate. Not only this, in addition to that, hormonal control also enters into this picture. Hormones, when epinephrine is released from the adrenal gland in response to stress, it triggers a series of events that suppresses the activity of glycogen synthase and at the same time stimulate that of glycogen phosphorylase. Because as I said, the two enzymes cannot be active at the same time. Otherwise, it will be a waste of chemical energy. Now, the activity of glycogen synthase is subject to the same type of covalent modification as a glycogen phosphorylase, but the response is opposite. Now, the inactive form is the phosphorylated form, and the active form is the unphosphorylated form. Now, the hormonal signals, glucagon or epinephrine, stimulate the phosphorylation of glycogen synthase via an enzyme called cyclic AMP-dependent protein kinase. Now becomes inactive after phosphorylation. At the same time, the hormonal signal is activating phosphorylase. Now glycogen synthase, the phosphorylation of this enzyme is more complicated in that multiple phosphorylation sites occur. As many as 9 different amino acid residues have been found to be phosphorylated. Now, glycogen synthase is also under allosteric control. And it is inhibited by ATP. This inhibition can be overcome by glucose 6-phosphate, an activator. So, ATP inhibitor, glucose 6-phosphate is an activator. Now, glycogen synthase, GS, exists in two forms. The phosphorylated or inactive form, known as GSD. D means dependent. Dependent on what? On high concentration of glucose 6-phosphate. Now, the non-phosphorylated or dephosphorylated is the active one, known as GSI, I يعني independent for glucose 6-phosphate. because it is active even with low concentrations of glucose 6-phosphate. Now, the fact that the two target enzymes, glycogen phosphorylase and glycogen synthase, are modified in the same way by the same enzymes links the opposing processes of synthesis and breakdown of glycogen even more intimately. Finally, modifying enzymes are themselves subject to covalent modification and allosteric control, and this is advantages to an organism. This control is advantage to the organism. Now we go, this is summary of this section. You can read it by yourself, the summary. And then we move to section 2 called gluconeogenesis, which is a process that produces glucose from pyruvate. Gluconeogenesis is the net synthesis of glucose from small non-carbohydrate precursors like pyruvate, lactate, glycerol, amino acid called alanine. Gluconeogenesis is not the exact reversal of glycolysis. Remember that in glycolysis we have 10 reactions. 7 of them are reversible. It's okay. They are the same enzymes are used in both processes, but 3 steps, step 1, step 2, and step 1 and 3 and 10 are irreversible, and these require new enzymes. So in order for gluconeogenesis to occur, we must reverse these 3 steps. You remember step 1, step 3, and step 10. So if we look here at this figure, if we look on it to the left here, from glucose all the way to pyruvate, this is glycolysis, which we talked about in chapter 17. Now, if we look from bottom to top, now we have gluconeogenesis. You see here the blue species in blue, green, and pink shaded boxes indicate other entry points for gluconeogenesis. For example, lactate, as I said, can be used. Amino acid alanine, glycerol enter from this point. So these are different entry points for gluconeogenesis. So let's begin this process called gluconeogenesis by reversing reaction number 10 in glycolysis. And it is the first step. Then pyruvate is carboxylated to oxaloacetate. This requires energy in the form of ATP, catalyzed by the enzyme pyruvate carboxylase. This enzyme is activated by acetyl-CoA, an allosteric factor, and requires biotin coenzyme. This biotin is a CO2 carrier in addition to a metal ion Mg+2. So all of these are required. For this first step, pyruvate is carboxylated to form oxaloacetate. This is in step 1. Remember, to synthesize 1 glucose, we must start with 2 pyruvate. So here, 2 ATP are required and 2 oxaloacetate are produced. Now, as I said, that all carboxylation reactions require biotin. And here you see the structure of biotin and its mode of attachment. This enzyme is pyruvate carboxylase. It has a lysine residue, and it forms an amide bond with the carboxyl group of biotin and the epsilon amino group of lysine residue. So biotin, a carrier of carbon dioxide, remember. Now the CO2 is attached to the biotin, which in turn is covalently bonded to the enzyme, and then the CO2 is shifted to pyruvate to form oxaloacetate. As we see in this mechanism, note that ATP is required for this reaction. So here you can see the mechanism. Look here how biotin is a carrier of CO2, and then biotin transfer this to pyruvate to generate oxaloacetate, the product, as you can see here. Now, the conversion of oxaloacetate in the next step to phosphoenol pyruvate is catalyzed by the enzyme called phosphoenol pyruvate carboxykinase, or اختصارا PPCK, which is found in the mitochondria and the cytosol. Notice here, oxaloacetate requires the reaction involves hydrolysis of GTP rather than ATP, but these GTP and ATP are equivalent. Oxaloacetate requires Mg+2 and the enzyme PPCK to produce phosphoenol pyruvate ZiO2. So here we have in step 1 carboxylation and in step 2 decarboxylation. Now, the successive carboxylation and decarboxylation reactions are both close to equilibrium. Now, once this oxaloacetate, that, if we go back here, this oxaloacetate that is formed in mitochondria can have two fates with respect to gluconeogenesis. What are these fates? Then it can continue to form PEP. The first fate can continue to form PEP, which can then leave the mitochondria via a specific transporter to continue gluconeogenesis in the cytosol. This is one fate. Now, the other possibility is that the oxaloacetate can be turned into malate via mitochondrial malate dehydrogenase, a reaction that uses an ADH. As we will see in the next figure, malate can then leave the mitochondria and have the reaction reversed by cytosolic malate dehydrogenase. Now, the reason for this two-step process is that oxaloacetate cannot leave the mitochondria, but malate can, because the mitochondrial membrane is impermeable to oxaloacetate. Now, you might wonder why these two paths exist to get PEP into the cytosol to continue gluconeogenesis. The answers brings us back to the familiar enzyme we saw in glycolysis, glyceraldehyde 3-phosphate dehydrogenase. Remember in chapter 17, the purpose of lactate dehydrogenase is to reduce pyruvate to lactate so that NADH would be oxidized to form NAD+, which is needed to continue glycolysis. So this reaction must be reversed in gluconeogenesis, and the cytosol has a low ratio of NADH to NAD+. كمان مرة. So the purpose of this roundabout way of getting oxaloacetate out of the mitochondria via malate dehydrogenase is to produce NADH in the cytosol so that gluconeogenesis can continue. Then the figure you see here is the mechanism by which oxaloacetate comes out of the mitochondria to the cytosol. This is called a compartmentalized reaction أولاً pyruvate is converted to oxaloacetate in the mitochondria, and then oxaloacetate cannot be transported across the mitochondrial membrane. It must be reduced to malate by NADH, and then malate is transported to the cytosol and then oxidized by this oxidizing agent NAD+, back to oxaloacetate to form NADH, and oxaloacetate is now ready for gluconeogenesis.